Duchenne muscular dystrophy is an X-linked recessive genetic disorder characterized by progressive muscle degeneration and weakness due to the alterations of a protein called dystrophin that helps keep muscle cells intact. Researchers have long pondered how to use exon-skipping therapies to skip over mutated exons that cause DMD in order to induce at least some dystrophin production. The MDX mouse is a valuable DMD model because it has a nonsense point mutation in exon 23 that leads to muscular dystrophy because of an absence of dystrophin. The MDX gene is also X-linked. A 1998 research paper led by Matthew Dunkley hypothesized whether or not a single-stranded complementary antisense oligonucleotide could be used at a post-transcriptional level to bind to the 3' splice site of intron 22. Antisense oligonucleotides are put in the polymer PEI to transfect into mice muscle cells, allowing it to enter the nucleus and bind to RNA about to be translated into protein. They used myoblast cultures. Dunkley disguised the splice site so that the splicing machinery skips over the mutated exon with a stop codon that inhibits dystrophin production. At 3, 24, and 48 hours post-transfection, cultures were washed twice in a phosphate-buffered saline and fixed in a 4% paraformaldehyde for 20 minutes for confocal microscopy. Laser confocal microscopy technique used exciting laser light to see if the oligonucleotide got inside the cell/nucleus in order to restore translation of the dystrophin protein. Box A represents 3 hours post-transfection, which is bright at the myoblast surface. B represents 24 hours discrete fluorescent patterns observed within myoblast nuclei. C represents fluorescent nuclei in myotubes. And D represents 48 hours fluorescence is localized to nucleoli of myotube nuclei. They also sent in antibodies that recognize specific dystrophin proteins that also fluoresce. Their results show that oligonucleotides got into the nucleus. A shows MDX mice without the treatment, and B shows MDX mice that received the oligonucleotide at intron 22, 3'. 1 to 2% of muscle cells were recognized to contain dystrophin at the sarcolemma. They demonstrated that complementary oligonucleotides, the 3' splice site of intron 22, can indeed induce skipping over the mutated exon 23 that contains a premature stop due to a nonsense point mutation. Antisense oligonucleotides can manipulate gene expression at the post-transcriptional phase. They also used buffers of forward and reverse primers for PCR sequencing, which revealed that RNA was spliced from exon 22 to 30, skipping the mutated exon 23 to allow dystrophin production to be in frame. This shows that even a little bit of dystrophin production could benefit DMD patients, so this method of exon skipping proved promising and has since been expanded on. A 2003 study done by Bianca Gebsky used a double-stranded partially complementary morpholino antisense oligonucleotide, complexed it with lipofectin, and transfected it as a lipocomplex into the nucleus. This is another promising study because they injected this into live mice and found that it restores some dystrophin in MDX muscle. In 2013, a group of researchers finally decided to do exon-skipping trials on human DMD patients. They hypothesized that the drug eteplirsen could skip over the mutated exon 51 to induce dystrophin production. Exon 51 had a deletion that prevented dystrophin from being produced. They had a sample size of 12 boys aged 7 to 13 with DMD and split them into 3 cohorts of 4 boys each. In the first cohort, 4 boys received 30 mg per kg of eteplirsen, and their muscles were biopsied after 24 and 48 weeks of treatment. In the second cohort, 4 boys received 50 mg per kg of eteplirsen, and their muscles were biopsied after 12 and 48 weeks of treatment. In the third cohort, 4 boys received placebo for the first 24 weeks and eteplirsen for the second 24 weeks. Their muscles were biopsied at 12 weeks and 48 weeks. One subgroup of 2 boys received 30 mg per kg of placebo and then 30 mg per kg of eteplirsen. The other subgroup of 2 boys received 50 mg per kg of placebo and then 50 mg per kg of eteplirsen. This graph shows that after at least 24 weeks of eteplirsen treatment, all cohorts had at least a 25% increase in dystrophin levels from baseline. Here is an image analysis showing increased fluorescent/stained dystrophin after specific times of treatment. This graph shows the functional efficacy of eteplirsen in a 6-minute ambulation test that investigates eteplirsen's effects on walking. The purple line shows the change from baseline in distance walked for the first 2 cohorts that received eteplirsen immediately at the start of the study. The gray line shows the change from baseline in distance walked for the third cohort that received placebo for the first 24 weeks and eteplirsen for the second 24 weeks. By 24 weeks, dystrophin levels had increased significantly in DMD muscle for all cohorts, indicating that duration of treatment may be more important for increased dystrophin production than dose size. Regardless of the small sample size that may reduce the confidence of the study's results due to variability in the sample, this study shows promising results for using exon-skipping technology to treat DMD in humans. Fortunately, the results of the studies such as these 3 I mentioned above have demonstrated results that are promising enough that the FDA approved eteplirsen as an antisense drug therapy for DMD in 2016 under the brand name Exon-51. Obviously, further research is needed to improve current exon-skipping technologies and discover new ones for other types of DMD mutations. However, these 3 studies have paved the way and laid the groundwork for the future of refining exon-skipping treatments for DMD. Directly before this was the bibliography. Thank you for your time.